Namaskar, my dear students. Hope you are all fine. Today we are talking about chapter respiration in the plants, and as already we have discussed the process and the definition and the types of the respiration, we shall be continuing after this, after the previous topics that we have discussed. And let me remind you what we have discussed in the previous lecture. We discussed that the process of respiration is the breakdown of the glucose into carbon dioxide plus water, and this is associated with the release of energy, and that energy is stored in the form of ATP and is used for the various processes of the life. We also discussed that the respiration takes place in the two ways, that is two types of respiration: the aerobic respiration, which takes place in the higher organism and it yields much more energy as compared to the anaerobic respiration, which takes place in the absence of oxygen and is found in the lower organisms, and in special instances it may take place in the higher organisms also. The glycolysis process is common in both, and then we discussed about the after discussing the anaerobic respiration, where one process which takes place generally in lower organisms, it is called as the alcoholic fermentation, where the end product is the ethanol, and in higher organisms where sometimes the anaerobic respiration may take place in addition to the aerobic respiration when the oxygen supply doesn't suffice with the need of the respiration, and that is called as the lactic acid fermentation. And in aerobic respiration we discussed that food or the glucose gets dissociated into pyruvic acid by the process of glycolysis, which gets converted into acetyl coenzyme A and enters into the Krebs cycle. After discussing the various steps of glycolysis, pyruvate conversion into acetyl coenzyme A and the Krebs cycle, now we have reached the next step, that is the electron transport chain. Electron transport chain is not the separate, but it is an inherent part of the above these processes, the Krebs cycle, and it takes place in the mitochondrial uh, space. Now, if you see this figure of the mitochondria, I am showing you the cut open portion of the mitochondria, and I have to tell you that mitochondria, as you can see, it consists of the two membranes, the outer membrane and the inner membrane, which is thrown out in the form of the folds. These folds are called as the cristae, which are having the structures on it to which we call F1 particle. The inner space, this one is called as the matrix, then the outer membrane and the inner membrane. Now the process of the respiration or the electron transport chain, it takes place in the inner mitochondrial space. Now how does this proceed, this electron transport chain? The chain is located in the inner mitochondrial membrane, and it consists of the various components of the electron transport chain. It includes cytochromes B, two types of cytochrome C, ubiquinone, flavoprotein FMN or FAD, as the case may be, then iron sulfur protein and enzyme cytochrome oxidase, which is intimately associated with the cytochrome A and A3. These components are arranged in four kinds of complexes, to which we respectively call complex 1, 2, 3 and 4. Complex 1 NADH dehydrogenase complex. Complex 2 is succinate dehydrogenase complex. Complex 3 is cytochrome BC1 complex, and complex 4, which is called as the cytochrome oxidase complex. There is a fifth complex, ATP synthetase complex, which is involved in ATP synthesis. Now the steps of the this electron transport chain, it proceed something like this. As you can see in this figure, I have shown you this the portion of the inner mitochondrial membrane. This is the portion of the inner mitochondrial membrane. This is the matrix, that is the inside, and this is the intermembranal space. Intermembranal space means the space between the inner membrane and the outer membrane. Now this space, how does the electron transport chain takes place? The metabolic pathway through which the electron passes from one carrier to another, that is called as the ETS, or the electron transport chain. Let's see how does it proceed. Electrons from NADH, here is the NADH. The electrons from NADH produced in the mitochondrial matrix during the Krebs cycle, to which we also call the citric acid cycle, are oxidized by NADH dehydrogenase. That is what I told you, complex 1, and the electrons are transferred to ubiquinone located in the inner membrane, that is here. It gets in the ubiquinone also receives reducing equivalents via complex 2, that is FADH2, that is generated during oxidation of succinate in citric acid cycle. You must remember that the electron transport chain is proceeding during the Krebs cycle, so the various substrates are being discussed. The reduced ubiquinone is then oxidized with the transfer of electron to cytochrome C. This ubiquinone, this is to cytochrome C via cytochrome Bc complex, which is what is complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and acts as a mobile carrier for the transfer of electrons between complex 3 and 4. The complex 4 refers to the cytochrome C. This is the cytochrome C. Now it contains cytochrome A and A3 and two copper centers. When the electron passes from one carrier to the next via complex 1 to 4 in the electron transport chain, they are coupled to ATP synthetase, to which we may call the fifth complex for the production of ATP, adenosine triphosphate, from adenosine diphosphate and inorganic phosphate. The number of ATP molecule synthesized depends on the nature of the electron donor, that is from where the electron has come. This is very important for you to understand that oxidation of one molecule of NADH gives rise to 3 molecules of ATP. What I mean to say is that is 1 NADH is equivalent to 3 ATPs. So while calculating the ATPs, you must take this fact into consideration. And on the other hand, 1 FADH2 produces 2 molecules of ATP. That is 1 FADH is equal to 2 ATPs. Although the aerobic process of respiration takes place only in presence of oxygen, the role of oxygen is limited to the terminal stage of the process. Yet the presence of oxygen is vital and cannot be ruled out, since it derives the whole process by removing hydrogen from the system. Oxygen acts as a final hydrogen acceptor, and wherein it ultimately forms the water. Let's see how does it occur. Unlike photophosphorylation, where it is the light energy that is utilized for the production of proton gradient, the respiration is different in that way that it is the energy of oxidation reduction utilized for the same process. It is for this reason that this is also called as the oxidative phosphorylation. Now let me discuss in little more detail the process of oxidative phosphorylation. So first of all, let us talk about the structure of the mitochondria. As I have already discussed with you that this process occurs in the mitochondria, and it is this space of the mitochondria, the matrix, and this is the F1 particles which play a very vital role. The mechanism of oxidative phosphorylation is a complicated aspect of the respiration, and three possible mechanisms have been put forth to explain the process. That is called as the chemical coupling. Now herein I have shown you that one cristae, and on the cristae are present the protein, the enzymes for the protein synthesis. This is one crista, this is the outer membrane, and this is one F1 F0 particle which is very important during the process of oxidative phosphorylation. So what I was telling you that three mechanisms have been proposed for this. One is called as a chemical coupling hypothesis, one is called as a conformational hypothesis, and third is called as the chemiosmotic hypothesis. What is chemical coupling hypothesis? It states that ATP formation takes place with the help of coupling factor, probably a protein there which is present and forms a high energy complex with one of the electron carrier, the CF carrier complex, whereas the third one is the conformational coupling hypothesis. According to this hypothesis, the mitochondrial membrane undergoes structural changes which induces the energy states or the conformations, that is the changes in the structure occurs. The conformation favors the release of energy which is used for ATP, catalyzed production of ATP from ADP and inorganic phosphate, that is ADP plus P gives ATP. This theory does not hold good because it lacks a little evidences, and what we have to discuss and which is most prevalent theory is the chemiosmotic coupling hypothesis, which was proposed by Peter Mitchell, and this theory is most widely accepted. And what this theory states that oxidative phosphorylation in the mitochondria is catalyzed by the multi-component enzyme complex called as the ATPase and are the coupling factor. ATPase has a small stalk and a headpiece located completely across the intermitochondrial membrane. You can see this portion is completely embedded in the inner mitochondrial membrane, and this is the head which is present towards the outer side of the towards the matrix of the mitochondrial membrane. The headpiece extends towards the matrix, whereas the stalk is embedded in the inner mitochondrial membrane and extends outwards the space between the outer and the inner membrane. The inner mitochondrial membrane shows a coupling mechanism and is permeable to water but impermeable to protons. Now you can see here that if it is impermeable to protons, where will protons be occurring now? They will be, and the respiratory chain is oriented in such a way that electron move in the inward direction and proton flow in the outward direction. Let me explain to you. The protons move in this direction and electrons move in this direction, as a result of which between the two membranes there is the proton rush. How does it occur? The Whenever the reduced NAD released from Krebs cycle enters the electron transport chain, three pairs of H plus ion, that is proton, across the inner mitochondrial membrane to the intermitochondrial space. They reach over here. For every NADH, three protons reach here. Similarly, NADH from the glycolysis and FADH from the Krebs cycle also transport a pair of hydrogen in the inner mitochondrial space. Each molecule of NADH released from the glycolysis or FADH from the Krebs cycle, two pairs of protons into the mitochondrial space are added. The unidirectional flow, because always the proton will flow in this direction, and what will happen, that unidirectional flow of protons to the outer side results in the accumulation of hydrogen protons in the intermembranous space. You can imagine that how much space is the, this is the inner intermembranous space. Now you can see in this figure that how much space is intermembranous space between the two membranes. It's quite lesser, and therefore the proton rush is created over here. The pH of the outer surface of the inner mitochondrial membrane lowers considerably due to concentration of net positive charge, because proton is a positive ion. Thus the pH gradient is generated, that is, gradient means difference in the pH. On one side it is positive, other side it is negative. The outward flowing or the pumping of hydrogen also generates a proton concentration gradient across the membrane. The pH gradient and the proton concentration gradient across the inner membrane builds a proton electrochemical gradient, that is proton motive force. The proton motive force derives proton across the membrane towards the mitochondrial matrix. Because the rush of protons is created, a proton gradient is created on this side, and so therefore it pushes the protons towards the inner mitochondrial space. The as the pH gradient is there what occurs, the proton motive force derives protons across the membranes towards the mitochondrial matrix. The return flow of proton down the electrochemical gradient through the enzymatic complex is responsible for the activation of ATPase, which derives synthesis of ATP from ADP. Therefore, there are the evidences to support that inflow of two protons through ATPase results in the synthesis of one molecule of ATP. Now, what happens when the two protons are push force towards the inner mitochondrial space? The ATPase enzyme gets activated, and ADP plus phosphorus ion, they result in the formation of ATP. For two protons there is one ATP which is formed. Now the ATP is formed at the head piece of ATP. This is the head piece. It is formed at the head piece of ATPase complex within the, which place is this? Matrix of the mitochondria, and then it is transported to the cytosol in exchange for incoming ADP, that is ADP from the cytoplasm. They keep on moving towards the mitochondria, and ATP are released in the cytosol, which are the energy currency of the cell. The net effect of respiratory chain ETS is that at least 3 ATP molecules are produced for each Krebs cycle. NADPH plus H plus ions, 2 ATP molecules are produced, and in the glycolysis 2 ATP molecules are produced for FADH2. Now we have seen that at various places the ATPs are produced, and in glycolysis there was the consumption of the energy also. So we would understand the total energy which is produced per cycle or per glucose molecule broken down if we make out a balance sheet. And here this figure is depicting how much is consumed and how much is liberated, and what is the net gain of the ATP. Now while calculating, you must remember that 1 NADH2 is responsible for 3 ATPs, because we will be talking about the energy produced in terms of ATPs only, and one FADH2 responsible for 2 ATP molecules. In glycolysis, 2 ATP, 2 NADH2. 2 NADH means 2 into 3, 6. So total number of ATPs obtained is 8. Similarly, oxidative decarboxylation of pyruvic acid yields 2 NADH, which are equal to 6 ATPs. And in Krebs cycle, 2 ATPs, 6 NADH, 2 FADH, which is equal to 24 ATPs, and in total you can see there are the 4 ATPs, and as a result of NADH2, which is equivalent to 1 is equivalent to 3, there are 30 ATPs plus 4 ATPs here, and almost 38 ATPs are formed per cycle of the aerobic respiration, starting from the glucose to the electron transport chain. So this gives us a idea that how much energy is produced and which is vital for the various life activities and is stored in the form of ATPs. So this is the complete respiratory cycle of the cellular respiration, but we must understand a few more terms, and first I will be talking about a very important term, that is respiratory quotient. What is this respiratory quotient? You must understand, respiratory quotient is the ratio of the volume of the carbon dioxide released to the volume of oxygen taken in the respiration in a given period of time at standard temperature and pressure, that is RQ. Respiratory quotient is equal to volume of carbon dioxide evolved or given out upon volume of oxygen absorbed or taken in. Now, respiratory quotient gives us an indication of respiratory substrate. We can calculate, if we know the respiratory quotient, we know which substrate is being used during the process of respiration. In anaerobic, respiratory quotient is greater than 1. And in aerobic respiration, if the substrate is carbohydrate, it is exactly 1, and if it is protein, it is approximately 0.9, and sometimes when the lipids are used as a respiratory substrate, it is respiratory quotient is 0.7. So it is very important to know the respiratory quotient, which gives us an indication as to which substrate is being used in the respiration. Now the next very important aspect that I am going to talk about is that many times we say that the respiration is the process which is the breakdown of the complex substances to simpler. In that context, we must say that respiration is a catabolic process, because metabolism consists of two processes: catabolism, breakdown, and anabolism, building up. Now, what is respiration? Catabolic or anabolic? In fact, it is, we will give a third term for the process of respiration, and that term is in between catabolism and anabolism, and that is amphibolic pathway. Respiration is the amphibolic pathway because it is both catabolic and anabolic in nature. I will make you understand how is it so. As stated earlier, that organic substances, carbohydrates, fats, proteins, whatsoever is the substrate, are broken down to release energy by the process of cellular respiration. This process is considered as a catabolism or catabolic pathway. However, the breakdown of the respiratory substrate provides carbon skeleton for the synthesis of large number of essential plant products. Which products? Polysaccharides, proteins, fats, nucleic acids, pigments, cytochromes, and various others. Thus, the same respiratory process which acts as a catabolic pathway for respiratory substrate, it also acts as an anabolic pathway for the synthesis of various intermediate metabolic products and the secondary metabolites. Thus, the respiratory pathway acts both as a catabolic and anabolic pathway. Thus, it's better to call it amphibolic pathway rather than as a catabolic pathway alone. So, that's all for the respiration. I hope you might have understood completely. So, in case you have any query or you have any question, you can anytime talk to me on my phone number, or you can leave a message at my email. So, my dear students, I suggest you to go through the process of respiration more than one times and learn the various intermediates of the reactions by writing down them, so that they become familiar and you understand the pathway. And so, for the complete understanding, this is must. And that's all for today. Thank you.